So today we're going to start with chapter 9 on patterns of inheritance. And we're going to look back for just a quick second on meiosis, in that remembering that meiosis and crossover that occurs in meiosis accounts for a large degree of variability in populations. Now, when we look at different populations, let's take humans, for example, we look at differences in hair color, eye color, height, weight, right, build, things like that. So some characteristics we can see, and then some we can't see. So certain characteristics of emotions, feelings, our cellular proteins and processes, right? We can't necessarily see those, but we can investigate them, we can look into them, and we can see patterns in populations. So chapter 9, where chapter 8 talked about cell division and how we take DNA and copy it and segregate that into different cells. Chapter 9 focuses on patterns of inheritance and how different traits in different organisms are passed on and expressed. So we know that when we duplicate and divide a cell, we are passing the DNA and all the cellular contents on to the next cell, the next two daughter cells. And when we do that, we're passing on traits, right? If you pass on DNA, you pass on the recipes for proteins, and you pass on traits. So we're going to talk about genetics. So modern genetics began somewhere around the late 1860s. This image shows Gregor Mendel, who is second from the right, holding, I think, is a pea plant in his hand, and they're kind of looking at him. And he started looking into genetics and inheritable traits using pea plants. He had no notions of chromosomes or anything like that. Chromosomes, genes, there were not even theories at this point. Of course, even before this, people had the idea that traits were passed on, right, from generation to generation or parent to offspring. But when Mendel published his paper in 1866, he stated that there were discrete heritable factors that retain their individuality when transmitted from generation to generation. Now, we know that discrete heritable factors are chromosomes. He didn't know that. But what he did know was that when he was doing his experiments with his pea plants, he found mathematics that told him that these pea plants had two, quote, heritable factors. Now, we know that that would be two chromosomes, right? Similar to the human diploid mechanism. So when he was studying his pea plants, what he would do is he would take the stamens and he would take the pollen and he would dust the carpel and he would make peas. And when he looked at those pea plants, he would look at various different characteristics. He would look at, do I have this somewhere? Here we go. Flower color, was it purple or white? Was the position of the flower on the side or on the end? Seeds yellow or green, seeds round, flat, or wrinkled, inflated or constricted pods, green or yellow pods, tall or dwarf in terms of how tall the plant was itself. So when he studied these 7 different traits, he always knew the parentage. He always knew what color or what he crossed from one to the other. And then he generated this plant with certain characteristics until he was sure that he had all true varieties. And then what he would do is he would start to mix them, right? So when he talked about a true purple, it means that there is no other color in that flower's history except purple. Same with the white. And when he crossed them together, right, he crossed a purple one with a white one, and he would assume that he would get either half and half purple and white, or he would get a lighter purple. But what he found out in that first generation, that F1, was that all the plants looked purple, just like the plants that were the parent in the P generation. So that was the first generation. But then when he took 2 of those purples from the F1 or the first generation, he crossed those and the white returned. In the F2 generation, he had some purple and some white. And how do you cross 2 purple flowers and get any white flowers? So this is him. He started doing it over and over and over and over again. And what he found, if you look all the way to the right on this slide, is his ratio. And for the most part, the ratio is going to be 3 to 1, from 3.15 to 2.84. But all in all, those numbers round down or up to 3. So 3 to 1. So not being a statistical geneticist or anything like that, he kept coming up with 3 to 1, 3 to 1, 3 to 1, meaning every time he crossed those, he would end up with what you see at the bottom, which is 3 purples and 1 white. So 3 to 1, 3 to 1, 3 to 1. Now, what does that tell him? That tells him that there are only two possibilities for each of these traits, either purple or white, nothing in the middle, either axial or terminal, nothing in the middle. So he started doing math, and what he found out, there were two heritable factors for each trait. And so this is where we get the modern genetics. This is where we start talking about dominant and recessive and things like that. Now, what does this mean? Right? If we look at the far right, we see a purple flower. All right, so how do we have a purple flower? Right, we have a purple flower if way over there on the left, there is a piece of DNA, CTAAA, TCGGT. If that piece of DNA is present on that chromosome, then it makes an enzyme that synthesizes purple pigment. On the bottom, the white flower, notice how the C is mutated or changed to an A, which means that the bottom gene segment, A, T, AAA, etc., does not code for purple, and so that equals the absence of purple, which is white. So this is just kind of giving you the idea of what does it mean when we say allele, right? An allele is where it is located on the gene, right? That locus for the flower color gene. You have an allele for purple or an allele for white, meaning that segment codes for purple or codes for white. Now, so this is sort of our continuum. We go from the gene to the protein to the purple. And we start getting dominant and recessive. So here we are. We're back to our purple and white, and we mix them up. We crossbreed them. And at the bottom in the F2, we end up with purple and white. So let's start talking about this. We have purple or white are the options. Now, in this case, purple is going to be dominant, right? Because it overshadows the white in the mix right there at the F1. So purple is dominant. We use the capital P to represent purple, the dominant condition, which is what you see top right. It says capital P, homozygous. Homozygous means they are two of the same purple genes for that plant. The lowercase p represents white or lack of purple. At the bottom right, you'll see two lowercase p's means homozygous recessive, meaning that white flower has two genes, both of them coding for white. In the middle, we have something called a heterozygote. It has one capital P and one lowercase p, or one dominant trait and one recessive trait. And since the dominant trait is purple, those flowers look purple. So on the left-hand side, you'll see something called phenotype. Now, phenotype is the expressed trait. So the flower is purple. That's its phenotype. The genotype is the genetic makeup expressed in letters. So that flower has two genes that code for flower color, and they are both capital. They are both dominant. The middle, the genotype, even though the flowers are purple, the genotype is heterozygote, meaning one chromosome contains a purple gene, one chromosome contains a white gene. And at the bottom, two white genes. So genotypes and phenotypes. The phenotype is the expression. The genotype is what do the chromosomes actually look like. So the new key terms here, dominant, recessive, homozygous, heterozygous, genotype, phenotype. So make sure you're understanding those 6 definitions pretty clearly. So now we're seeing something in the middle. It's called a Punnett square. You may have seen a Punnett square before. We don't know. The Punnett square is a good way to be able to cross genotypes to see what the resulting genotypes could be. This is a little bit extra. On the right-hand side, you'll see a 16 square. That's if you want to cross 2 traits, which we won't get into. But either way, notice the numbers. It says phenotypic ratio 3:1. Phenotypic ratio 9:3, 3:1 means you basically multiplied 3:1 2 times by each other. You squared it. So 3:1 is the overarching variable here, or the common constant when we're talking about having 2 chromosomes. This is a picture of labs, black labs. And black labs are have a capital B, and chocolate labs have a lowercase b. Normal vision, capital N. blind, lowercase n. So it's just kind of showing you mixing up the double heterozygotes and what you would get from that. Um, so again, these are just sort of practice examples. These are very complicated, and so is this. This is more along the lines of what you would be asked to do a Punnett square to cross the two genes. I wouldn't ask you to do a 4, but it's just giving you an example. So here we go. So the genotype of the black dog, right? We know it has one capital B because it's black, but we don't know what the second one is. It could be a lowercase b, or it could be a capital B. If we cross that black dog with a brown dog, and then we have to see what the puppies look like. If every single one of the puppies is black, then more than likely the black dog has 2 capital B's in its genotype, or it is a homozygous dominant. If some of the puppies are chocolate colored, it must mean that the black dog is a heterozygote, right, in order for it to have a lowercase b to donate to its offspring. So we want to kind of get an idea that each chromosome contains an allele for a different characteristic, and each characteristic for what we're going to discuss is a result of 2 genes. So you can have the homozygous dominant, the heterozygote, or the homozygous recessive. Now remember, we have 2 copies of each gene for the humans, for these dogs here. One set comes from the female parent, one set comes from the male parent, right? We're talking about inheriting chromosomes, and those chromosomes leading to certain traits. So a cross between a homozygous purple flower, which is 2 capital P's, and a homozygous white flower, which is 2 lowercase p's, will result in offspring with purple flowers. This demonstrates what? Because the purple flowers of the offspring are going to have 1 capital P and 1 lowercase p. So this must be demonstrating C dominance. The purple is going to overshadow the white. For this one, what you'd want to do is do a Punnett square between DADB and DCDD. And if you were to do that, you would know that we would end up with ACAD. The letter A is going to be the correct answer. And I could say AD, AC, AD, AD 50 times. But what I would recommend you doing is just do the Punnett square, crossing the A, B, C, D's, and you will get letter A. Listening to me explain it to you with the C's and the D's and the D's and the superscripts is going to be complicated. All right, so now we're talking about characteristics in pea plants, right, for the most part. But the book also talks about birds, but these rules also apply to human traits. So things like tongue rolling, if you're able to roll your tongue, or freckles, hairline, earlobe attachment. This picture here are 2 sisters that are, one has a widow's peak and one has a straight hairline, and talking about that, which we'll do in a second. So this is, what I want to bring up now, though, is often recessive traits are the more prevalent one in a population. Freckles are dominant, but most people have no freckles, right? Straight hairline is recessive. Most people have a straight hairline. Very few people have a widow's peak. So when we talk about traits, don't assume that dominance equals prevalence. It doesn't. Those 2 things have nothing to do with each other. It really depends on which one is more beneficial to the population at a given time. So don't assume that dominance equals prevalence, right? That's not a thing. So there are over 100 human genetic disorders that are controlled by a different gene, which we'll talk about in a minute. But this is what we're going to call a pedigree chart. A circle equals a female, a square equals a male. If the lines are like on the top left, Al and Beth. Al and Beth procreated or had children and had those 4 children on the bottom, Evelyn, Frank, Gary, and Henry. Now, there are squares and there are circles. If the square or the circle is filled in, right, then it is the straight. If it is hollow or open, then it is the widow's peak. And so you can follow that down. Al and Beth both had widow's peaks. 2 of their kids did, 2 of their kids didn't. On the other side, Charles and Debbie, one had a widow's peak, one did not. The children, one had a widow's peak and one did not. When Henry and Isabel got together and had 2 children, Kristen and Lori, Kristen did not have a widow's peak, straight hairline, inherited the 2 recessive traits from her parents, and Lori is either homozygous dominant or heterozygote. We're not really sure. We'd have to see if Lori had children, what her children would be, and if we could figure it out from there. But so here's just sort of following through the generations, the different genotypes accompanying their phenotypes. So pedigree charts are pretty neat way to look at different traits as they pass through a family. So here we have some autosomal disorders, recessive and dominant disorders in humans, albinism, cystic fibrosis, sickle cell disease are going to be sort of our common or more prevalent recessive disorders. Achondroplasia, Huntington's disease, hypercholesterolemia are going to be some of our more prevalent dominant disorders. And again, a recessive disorder means you have to have two, you have to be the heterozygous, homozygous recessive to inherit that. Here, in the dominant disorders, you may need to be both capital and lowercase, meaning the heterozygote, or you could have two be the homozygous dominant. So here we have freckles and no freckles, right? No freckles is the more common, and freckles is the less common. On the bottom, albinism is obviously the less common trait, but it is, and then on the right, on the left, normal pigmentation, right, is the dominant. So it doesn't matter. Sometimes the dominant trait is more prevalent. Sometimes the recessive trait is more prevalent, right? So it's not about dominant equals prevalent. That's not a thing. So when we talk about different recessive disorders, again, you need to have the two, the homozygous recessive usually as a result of heterozygous parents. This is a little question about a child who's suffering from Tay-Sachs disease. Neither parent has Tay-Sachs, nor does anyone in their family, right? But the child does. Tay-Sachs is a recessive disorder. So the answer here, right, is C. If you were to do a Punnett square of two heterozygous parents resulting in a homozygous recessive child who had Tay-Sachs disease, you would see that that results in a 25% chance. Now, what happened to this child has no bearing on what happens to another child, still 25% chance. So the answer here is C. This is a physician that has achondroplasia, right? And you can see here that the achondroplasia is a dominant disorder, right? It's a form of dwarfism. It's a heterozygous condition. So you have to be a heterozygote to inherit achondroplasia. Huntington's disease is also a dominant disorder. So the grandfather has Huntington's. His daughter has a 2-year-old. No one else in the family has the disease. So it seems as though the daughter is probably a heterozygote, and she has a 2-year-old. If she's a heterozygote, anywhere between 0 and 50%, right, that the daughter will contract the disease, because more than likely, 50% chance that the dad passed down the gene to her. So about 50% chance. This opens up a whole other can of worms. What happens if you're the daughter and now you have a baby and you want to have a second child? Now you're 50% chance that you have Huntington's disease, which is a lethal nervous system degenerative disease, right? These are interesting questions. Right? Do you get genetic testing? Do you choose to have a baby, not have a baby, right? These types of pieces of information can really be helpful, but they can also bring up really, really complicated questions. All right, so we just spent a little bit of time discussing heritable traits that are passed on via a single allele or a single locus. However, most characteristics are inherited in ways that follow more complex patterns. These are the complex inheritance patterns that we're going to talk about now. So when we talked about Mendel's peas, there were 3 different genotypes, right? We had the homozygous dominant, the heterozygote, and the homozygous recessive. But we only had 2 phenotypes, purple and white. Sometimes that heterozygote in the middle is going to be its own thing. That's what you see here. These are snapdragons. Snapdragons exhibit the following effect called incomplete dominance, meaning the red is incompletely dominant over the white. They blend together. So when red snapdragons are bred with red snapdragons, they're all red. When red snapdragons are bred with white snapdragons, they are all pink, right? So pink is an intermediate effect, right? One allele is not completely dominant in the heterozygote. Heterozygote exhibits an intermediate characteristic. So intermediate between red and white is pink. So this is incomplete dominance. The next type of complex inheritance pattern that we're going to talk about is blood typing. Now, in humans, normal human blood type, we have 4. And we have either type A, type B, type AB, or type O. And you can see at type AB that A is not dominant over B and B is not dominant over A. They are co-dominant together. So when you look at the genotypes, you don't just see A's and B's, you see a letter I. I means the presence of a protein or a carbohydrate, and lowercase I means the absence of that carbohydrate. Now, when we talk about blood typing, we have 2 possible carbohydrates. We have carbohydrate A and we have carbohydrate B. Carbohydrate A gives you A blood. Carbohydrate B gives you B blood. If you have A and B, that's the co-dominant condition, and you have type AB. If you have neither A nor B carbohydrates, you have type O blood. So this is a co-dominance situation. What about this one? All right, this is skin tone. This is called polygenic inheritance. You'll notice that there are 3 genes here, the A's, the B's, and the C's. Now, this is a fictitious example because, for the most part, skin color is affected by multiple genes, but we're going to go with 3, A's, B's, and C's. And you'll notice how from top left to bottom right, skin tone goes from lighter to darker, depending on how many dominant conditions you have. All right, so it's polygenic. Multiple genes affect a single characteristic. Right, so your skin tone is your skin tone based on a variety of different genes. All right, so this is polygenic inheritance. This one over here, I know we just looked at labs, but we looked at, where's my sickle cell? Oh, I guess I didn't put it. Before we do that, we're going to talk about pleiotropy. There's not a slide for it, unfortunately. I don't know why, but pleiotropy means there are different phenotypic characteristics, but one gene. So the one gene that gives somebody sickle cell may affect different people in different ways. If I have sickle cell, I might have a lot of pain, kidney damage, blindness. Someone else has sickle cell, they might have trouble breathing, they might have tingling and numbness. So different phenotypic characteristics as a result of one genotype, and that's going to be pleiotropy. This picture right here of the black labs is called epistasis. Epistasis is when a gene at one location alters the expression of a gene at another. So when we look at the black labs, what do they all have in common? They all have at least one capital B. When we look at the yellow labs, or the black and brown labs, what do they all have in common? The black and brown ones all have at least one capital letter E. When we look at the yellow labs, you'll notice that they never have a capital E. They may have a capital B, but the E is what determines whether there's going to be color. So if there's a capital E, then the color B will be expressed. If there's no capital E, then no color will be expressed. So you'll notice the second one down on the far right column where it's lowercase b, so the homozygous recessive for color, but the heterozygote for whether it has color or not. So it has a capital E, but it has 2 lowercase b's, so it's brown. Just below that, It has a capital B, but it has 2 lowercase e's. So it's not going to have any color. All right, so lowercase e means no color. That's how you get different colors for Labrador retrievers. Kind of an interesting little factoid there. This right here is called a hydrangea. It's a plant. Most of the hydrangeas we have in the Northeast are going to be blue, but it's the same plant, right? So what is this? This is called a multifactorial trait. This is when the phenotype is affected or determined by some environmental condition. For example, nutrition influences health, growth, body condition. Exercise can alter your body's shape. In this case, it's soil pH that tells whether the hydrangea is going to be blue or whether it's going to be pink. If you have hydrangeas in your yard and you alter the pH of the water that you water it with, you can change the color of the hydrangea from blue to pink and back again. It's fascinating. So it's a multifactorial trait. It's not really the genetics, it's the environmental influence on those genetics. So when a disease is said to have a multifactorial basis, it means that A, both genetic and environmental factors contribute to the disease. This is perfect little chart here because it tells you of the complex inheritance patterns and it tells you the description of each one, going all the way from codominance to multiple alleles to pleiotropy to epistasis, polygenic inheritance. All right, so this is a great table to sort of have nearby. Now, what we want to remember is that when we're talking about traits, traits come on chromosomes. So this is now giving yellow and green seeds as it passes through the generations and showing you the phenotypes when they come out at the bottom. It's sort of correlating meiosis with the genetics, so you can see what we're talking about. And lastly, one sort of different type of inheritance pattern is called X-linked. These are called sex-linked genes and traits. That means that there is a gene on either the X or the Y chromosome that is more prevalent in one gender. For example, if it's on the Y chromosome, then mostly biological males are going to, it's only going to affect biological males, right? The X chromosome or X-linked traits can affect both males and females. Some of our X-linked traits in humans, color blindness, hemophilia, Duchenne's muscular dystrophy, there's a variety of different X-linked diseases and disorders. This last little graphic here shows just different ways in which organisms can pass on their genes. We have the XY system. There's XO, ZW, haplodiploid. You're not responsible for any of this. I was just sort of showing it to you. We've reviewed non-disjunction already, so we're not going to do that again. As you can see, this young lady has Down syndrome or trisomy 21, and you can see her karyotype here in the middle at the bottom. You'll see 3 of chromosome 21s, right? And that is as a result of a non-disjunction event from one of her parents. So now she has 3 chromosome 21s, and it's a female because you can see how there's an X, 2 X chromosomes. We talked about different ways during replication that genes can be altered, deletions, duplications, inversions, or translocations. So that's the end of chapter 9. Again, it sort of overlays the phenotypic or expression of traits with the stuff we talked about in meiosis, and hopefully you can see those connections. We're going to sort of really get into that in chapters 10 and 11. How do we get from gene to chromosome?